Thank you.
Perfect. Thank you. Thank you so much. And thank you so much for the introduction as well as the invitation. So something I neglected to mention in my bio was that I did my PhD at the University of British Columbia. And while I have many fond memories of Vancouver, until today, none of them included low moisture foods. So thank you for this opportunity. So the goal of my talk today is for you to have a little bit of an appreciation of the food safety risks associated with low moisture foods. After a bit of an introduction, I'll go into three projects that we're currently working on and with some conclusions, and hopefully there will be time for some discussion at the end. So in Canada, food safety is a shared responsibility between members of the food industry, consumers, as well as all levels of government, with interventions from farm to fork. At the federal level, there's four major players. We all have our distinct roles with respect to policy and actions, but we often work together. So for example, during outbreak investigations, those investigations are led by the Public Health Agency of Canada, and the Canadian Food Inspection Agency will lead the food safety investigations and the corresponding actions, such as food recalls. At Health Canada, I like to say that our work begins before and after a foodborne outbreak. So in the Bureau of Microbial Hazards, we have a research division with expertise in various areas of food microbiology. And the goal of our research is to inform food policy standards and guidelines so that we can prevent food safety incidents from occurring. And this slide just shows some examples of some of our outputs that are available on canada.ca/food safety. So what is Salmonella? The very simple answer to that is it's a bacteria that can cause disease. It can make people sick. And it's very good at doing that. And it's been doing that for thousands of years. So this is what Salmonella looks like under the light microscope and on an agar plate. So the actual bacterium itself is a rod shape, and its surface is covered by a sugar molecule called LPS, which you can see on this bottom electron micrograph. It's this fuzzy line here. And LPS stands, in case I didn't mention it, it stands for lipopolysaccharide. So the surface of the cell is also covered in these short hair-like appendages called fimbria, and the fimbria are responsible for adhesion to various surface and other cells. And we can see this adhesion in the lab in the form of a structure called a biofilm, which is shown here. The cell also has longer hair-like appendages called flagella, and flagella help the bacteria move around. And we can see this in the lab on motility agar. Salmonella has unique metabolism, and we take advantage of that metabolism when we're trying to detect it from food. So these two bottom plates here show some of the color reactions that Salmonella can give, and this is how we can distinguish Salmonella from other bacteria that are in our food. Salmonella is also a genus, and the genus is composed of two different species. We have Salmonella bongari and Salmonella enterica. And Salmonella enterica is divided into 6 different subspecies. And over 99% of human illnesses are caused by subspecies enterica. So all Salmonella subspecies and species can be further broken down into serological variants or serovars, depending on the type of LPS and flagella that they have on their surface. And in the Salmonella world, we have lots of fun because all of the serovars have names, and you'll be hearing some of those names throughout the talk. So in Canada, in 2023, there were 6,281 reported cases of salmonellosis, and approximately 70% of those cases were attributed to 10 different serovars, and the remaining 30% were attributed to over 100 different serovars. And the important thing to remember when you look at Salmonella case numbers is that the case numbers are an underestimation of the real world situation. And this is because most people who become sick with salmonellosis, they'll get better on their own without seeking medical care. So we never see those cases. And the Public Health Agency of Canada has done some work to show that for every one case that's reported to them, there are 25 additional cases in the community. So when you see a case number of Salmonella, if you multiply it by 26, you'll get a better estimate of the real burden of illness. And you can see that it is quite significant in this country. So low moisture foods are a really broad category of foods. I've shown some pictures here on the slide. And the thing that unites low moisture foods is they have low levels of available water. And so water for bacteria to use to metabolize. And pure water has a water activity of 1, and Salmonella needs to have a water activity of 0.94 or above to grow. The technical definition for a low moisture food is anything below a water activity of 0.85. And this is because foodborne bacteria, many of them cannot grow beyond this point. At a water activity of 0.6, you see all microbial activity ceases. So this includes things like yeasts and molds. So low moisture foods present some unique risks compared to other types of food. So there are some low moisture foods that have very long and convoluted supply chains that can involve multiple countries in terms of where things are grown, where they're produced, and where they're distributed and consumed. And it's really hard to maintain hygiene over those long distances with so many different players. The long shelf life of many low moisture foods means that there's opportunities for multiple exposures over the shelf life of the contaminated food. Because of their nature, you can't use water in low moisture food processing facilities. And so dry cleaning methods such as vacuuming and brushing, they're not as effective as using water and water-based soaps and sanitizers. And third, with low moisture foods, there's a general lack of validated kill steps. So these are the procedures that have actually been shown to reduce levels of pathogen in a particular food matrix. And you can't really generalize from one food type to another because we know that the food environment can affect heat susceptibility. So for example, if you have a food that's quite high fat, it's going to have a different heating profile than a food that has less fat. And in this panel here, I've shown how Salmonella can respond to heat. So the first panel here is just normal Salmonella. So the top photo is unheated Salmonella. And after 10 minutes at 80 degrees Celsius, you can start to see some signs of heat damage. So these black dots are protein aggregates. And after 1 hour at 80 degrees Celsius, you start to see the DNA aggregating. When Salmonella is in an environment of water activity 0.3, you can see some changes in the membrane, but these aren't lethal. What's more important is to look at the cytoplasm here. So after 10 minutes and 1 hour, you can see there's really no difference and there are no visible signs of heat damage. So the consequence of having Salmonella in these low moisture foods is if people consume them, they become sick. And if more than one person becomes sick from eating the same food, we then have an outbreak. So these are just some selected outbreaks I've chosen from the literature just to show you that we've been dealing with this problem for quite a long time. And it's involved a variety of different vehicles, food vehicles, all over the world with a variety of different Salmonella serovars and with some astonishingly high case numbers too. So I'll direct your attention here to 2022. Some of you may remember around Easter time, there was an outbreak related to chocolate. There's a lot of chocolate recalls in Canada at the time. So this outbreak originated from one production facility in Europe, and the product that was implicated was distributed to 113 countries, and 11 countries had illnesses. So this just shows you the scope of the public health and economic costs of having salmonella in low moisture foods. So the first commodity I want to talk about is sprouted chia seed powder. And in 2014, there was an outbreak in Canada and the United States linked to the presence of salmonella in these powders. And at the time, this was a new type of food that many people hadn't have heard outside of this outbreak. And this food has a very interesting production process. So the seeds are first sprouted, the sprouts are dried, and then the dried sprouts are ground into a powder, which can then be sprinkled onto smoothies or yogurts, things like that. And the sprouting step is the step that is interesting microbiologically because the same conditions that promote sprouting are the ones that can promote bacterial growth. So we were really curious to know what the levels of salmonella were in this food product. We were also interested in knowing more about this food. How can salmonella survive? Can salmonella grow in this food? And if salmonella is surviving, how is it doing that? So we were able to obtain some of the contaminated samples from our colleagues at the CFIA, and we wanted to first see what the levels of salmonella are. So we first tried direct plating, and direct plating involves making a homogenate of the food in a broth, and then you plate on agar, you count the colonies, and then you can calculate the levels of salmonella. So when we did this, we didn't get any results. And then we thought about it for a bit, and then we reasoned that the lowest level you can detect using direct plating is 10 colony forming units per gram. So we thought, well, maybe there was less salmonella in the food. So then we turned to our detection method, and our detection method is really good. It can detect if there's one cell present in the food, we can detect it. So what we ended up doing was we took our entire sample and we divided it into 15, and we did 15 detections simultaneously in parallel. And by looking at the pattern of the positives in the samples, we can then consult our statistical tables to calculate a most probable value for the amount of salmonella that's going to be in these products. So with those 6 products, we found levels ranging from 0.2 to 35 cells per 100 grams. So really, really low levels. And so our MPN method from the previous slide, it's very sensitive, but it takes 4 days to get a result, and you need to have a lot of sample. So we were wondering if there was a way that we could distinguish the salmonella contaminated samples from samples that were not contaminated. So we did a comparison of salmonella positive samples and salmonella negative samples, trying to see if we could find some sort of indicator of contamination. And we first looked at physical chemical properties of the food. So that's shown here in this graph on the top half. So we looked at pH, we looked at water activity, and we looked at moisture. The salmonella negative samples are shown in orange, and the positive samples are shown in blue. And you can see with the physical chemical properties we looked at, the samples overlap quite a bit. So we couldn't really distinguish them using PhysChem. So then we started to look at the microbiology. And what we found was there are 2 groups of bacteria, the Enterobacteriaceae and the coliforms. They had significantly higher levels of those were found in the salmonella positive food samples compared to the salmonella negative ones. And the beauty of this method is you only require 25 grams of sample and you get your result within 1 day. So this result showed us that perhaps we can use the levels of Enterobacteriaceae and coliforms to monitor the hygiene of the food product as well as the food production process. So we did the initial enumeration of the Salmonella positive food samples in 2015, and that's shown here on this y-axis of this graph. And we were curious to know how long can Salmonella survive in these environments. So we checked the levels in 2020, and then just this year, it was 10 years that we had these samples in the lab. So we checked again, and we were able to pull out Salmonella from all 3 time points. So this shows that Salmonella can survive for at least 10 years in this product, which is really astonishing to me. So then the next question becomes, how is it surviving for so long? And what mechanisms, what adaptations is the bacteria using? So to answer this question, we chose to compare strains that were isolated from the food sample. So that's what OB is on these slides. It stands for outbreak. And then we compared it to some lab strains that we had in our culture collection. So LB means lab. And we tested one strain from 2 different serovars, Newport and Bredni. And we ran them through a variety of phenotypic tests. And we found that the outbreak strains have unique properties. So if you look at this top left panel here, you can see that the Newport outbreak strain and the Bredni outbreak strain, they form more robust biofilms than the lab strains. And this is shown in these panels of pictures. It's just showing biofilm formation in different ways. So every single assay we had for biofilm formation, the lab strains did not form any, whereas the outbreak strains formed really beautiful ones. The outbreak strains also could grow in hyperosmotic conditions, so really high concentrations of sucrose. This curve is showing the outbreak strains grown in sucrose is pretty comparable to our regular media, whereas the 2 lab strains, they kind of struggled in the sucrose condition. The outbreak strains also exhibited desiccation resistance compared to the lab strains. So we hypothesized that the ability to withstand these stresses and to form biofilms, perhaps that contributed to the survival in the seed powders. And we tested this by inoculating each of the 4 strains in clean sprouted chia seed powder and assessing the survival over 6 months. And we were very happy to see that the outbreak strains, they did out-survive the 2 lab strains. So some key takeaways from our sprouted seed work is that these powders can have really low levels of Salmonella. And what this means for detection is we might need to go beyond our standard sample size in order to increase the sensitivity of our methods so we can have a better increased probability of detection. And the use of enumeration of hygiene indicators can give information on the acceptability of the food. And our work with the Salmonella strains has shown us that all strains of a serovar are not equal, and low moisture food adapted strains, they can they can present some challenges for removal because we saw that they form biofilms. And when doing challenge studies, the choice of strain is really important. So now I'm going to switch over to spices. And this table shows some of the public health impacts of having Salmonella in spices. So again, it's a very long-standing problem with a variety of different spices being implicated. with a variety of different Salmonella serovirus being involved all over the world. And the issue with spices is that it's really hard to detect Salmonella in spices. Um, Spices have antimicrobial compounds that can inhibit the bacteria that you're trying to detect. And the low moisture environment doesn't help because even though bacteria are surviving in low moisture conditions, they're not at their peak. So they're going to be injured. And then the antimicrobial in the spice just exacerbates, the injury just exacerbates the antimicrobial activities. Also, spices have other issues with them that complicate their handling. For example, they can absorb water. Sometimes the color can interfere with our color reactions in the lab. So just a very difficult commodity in general. So what we wanted to do with this project was we wanted to, oh, excuse me. So in our method, we recognize four spices as being inhibitory. So oregano, allspice, and cinnamon, and cloves. And the way we get around these detection issues is we will do an additional dilution of the spices in order to dilute out the antimicrobial compounds. So oregano, allspice, and cinnamon require a 1 to 100 dilution, and cloves require a 1 to 1,000 dilution. So what this means for our method is with our two-class sampling plan using a sample size of 25 grams, you'll need at least 12.5 liters of our primary enrichment broth, which we call BPW, for oregano, allspice, and cinnamon. And for cloves, you'll need 125 liters per sample. So that's a lot. And knowing what I just told you about the low levels of Salmonella, spices also tend to have lower levels. So that 25-gram sample is not going to be sufficient. So it's just a really massive undertaking that a lot of labs probably don't have the capacity to do. So we set out to see if we could improve our method. So our aim was to see if we could just reduce the volume required to detect Salmonella in allspice. And we tried a variety of different methods, which are shown here on the bottom. So this molecule here is eugenol, which is the predominant antimicrobial compound in allspice. So our reference method recommends a dilution of 1 to 100. We wanted to see if we could just limit the, for lack of a better word, the contact time that the bacteria has with the eugenol. So our first method involved just very quickly adding the broth to the spice, shaking it and decanting it and doing the detection on the broth. The second method used corn oil to see if we could partition the eugenol out of the vicinity of the bacteria in the broth. The third method used a compound, a silica-based compound called beta zeolite to see if we could just pull that we could pull that compound out of solution. And then we also looked at neutralizing agents in phase 2 of the experiment. So we decided to use allspice. Allspice comes in two forms. You can have the whole dried berries, or you can have the ground allspice. So we looked at both, and we wanted to reflect the diversity of Salmonella. So we did a survival study with 10 different strains, which is shown here. And what we saw was that there were strain to strain variations in the rate of dying off. So we decided instead of using a cocktail for subsequent experiments, we would just look at individual strains. So we chose the 4 strains that are highlighted in green for the rest of our experiments. So this slide shows the results of phase 1. So we tested tifamurium with whole berries. The reference method is colored in orange and our experimental methods are colored in green. And with Tifamurium, there was really no difference between the methods in terms of recovery. With Tennessee, we did see differences. Um, it looked like the reference method and corn oil were the two best methods for Tennessee. For ground allspice, we used Munchin, and similar to Tifamurium, the five methods were pretty comparable. With Seftonburg, we did see some differences. And again, it was the reference method and the corn oil that were the top two methods that we tested. So for phase two, we wanted to see if we could use inhibitory agents to kind of block the action of the eugenol. So the first inhibitory agent we used was casein because it's already used in our method. So casein is a milk protein, and we use it when we want to detect salmonella in chocolate because it's thought that the casein will inactivate those anthocyanins in the chocolate. Next, we used potassium sulfite because, again, it's used in our method to neutralize the compounds in dried garlic and dried onion. So our method uses potassium sulfite in tryptic soy broth, but I like to keep things simple. So I wanted to see if the base broth makes a difference because we use BPW for everything else in the method. So we added potassium sulfite to BPW. And the last inhibitor was something that we had read about. It's used quite commonly in pharmaceutical methods, and it's a combination of tween, lecithin, and histidine. So this slide here shows the results of phase two. So again, with the Tefimurium, it performed really well with all five methods. With the Tennessee, we did see quite a bit of variation. But the method that pulled the most salmonella out of the allspice was the one with the three inhibitors, um, the tween, the lecithin, and the histidine. In the ground allspice, with the Munchin, we got really good recovery with all five methods, but it looked like the TLH was the best one. And with Seftonburg, again, it looked like all methods, with the exception of the method using the casein, did really, really well. So if you'll remember from phase one, our best method was the corn oil, and in phase two, it was this TLH. So then we did a comparison of corn oil and TLH just it just edged out corn oil as being better at pulling out salmonella. So we wanted to do a proper validation study in the context of our method. So we compared our reference method, so the 1 in 100 dilution, with our alternative method, which was TLH. And if any of you are curious, this is what a 1 in 100 dilution of allspice looks like in the lab. And this is what 5 kilos of allspice looks like. And we did these experiments over Christmas, so it was very festive. So our validation study consisted of 3 trials using 20 samples of for each method. And we inoculated at really low levels to try to mimic those low levels we would see in the real world. So our reference method, we were able to detect salmonella 33 times out of 60. And with our alternative method, we were able to detect 42 times out of 60. So this was a result that made us very happy, and we are continuing validating more spices to see if this broth can be a more general purpose broth for spices. So as happy as we are with our culture method, by using this combination of tween, lecithin, and histidine, we still are having issues with molecular detection of salmonella using PCR, so we're still working on that. And another takeaway we have from this work is that the method performance, it really varies from strain to strain. So again, the choice of strain is really important when you want to validate a method. So the last commodity I want to talk about is tahini. And tahini is it's a sesame seed paste or a butter. It's made from ground up sesame seeds. And it's a staple of Middle Eastern cuisine. And you can see on this table here, again, it's been very problematic for a long time, um, all over the world with different Salmonella cerevars being implicated. And tahini is what I like to call a global commodity. So it's the sesame seeds are, for the large part, grown in various countries in Africa and in Asia. And those seeds are imported into the Middle East, various countries. And here is where the seeds are processed into tahini and then distributed all over the world. So these yellow arrows show places where outbreaks have been reported related to tahini. So for our research, we really wanted it um, to make an impact where we could have some influence. And so for us, that was here at the consumer level, so right at the point before consumption. So we did a little research and we consulted our friend, the internet, and we found that one of the ways that tahini is consumed in this country is through sauces. So these are usually added to sandwiches or salads right after as the final step before they're being prepared and right before they're going to be eaten. And there's various recipes for tahini sauce. The most basic one is just diluting it in water. You can also have recipes that incorporate lemon, garlic, and other spices. So we wanted to understand how hydration is influencing the levels of salmonella, should it be present in tahini, and whether or not these recipe ingredients, because we know that lemon and garlic are antimicrobial, we wanted to know whether those ingredients could influence the levels of salmonella. Also, we wanted to see what is the impact of storing these sauces at room temperature versus 4 degrees in the fridge. So our 4 recipes involved first just diluting the tahini in water. So that would raise its water activity. We wanted to see what a sauce looked like where the water activity was maintained as low. So we diluted the tahini in oil. We added various concentrations of lemon juice. So that was adding an acidic element to our sauce. And then we added garlic as a source of antimicrobials. So this slide here shows a summary of our results. So this first column here shows the various tahini sauces that were stored at room temperature. And the second column here shows the tahini sauces that were stored in the fridge. So the first takeaway is that regardless of the ingredients used in the sauces, storing them in the fridge, we did not see any replication of salmonella at all. Room temperature, the situation was quite different. So we saw growth in 2 conditions. The first condition was when we increased the water activity by adding water, shown here in this black line. When we added water and garlic, we also saw growth. So it didn't look like it didn't look like garlic was doing anything in our system. When we maintained the low water activity by adding oil to our sauce, we did not see any replication. When we added lemon juice at a final concentration of 25%, that's shown here on this bottom graph, this green line, we didn't see any replication. And this yellow line shows lemon juice with garlic. So again, garlic has no effect. When we increased the lemon juice concentration to 50%, we not only did we not see replication, we actually saw a decrease in salmonella, um, both at room temperature and also in the fridge. Um, so these results tell us that, but when you increase the water activity of tahini, you're also increasing the risk of salmonella replication. But that replication can be mitigated by adding acids to your tahini sauce and storing it in the fridge. So our optimal recipe for tahini is half a cup of tahini, half a cup of lemon juice, plus or minus garlic, mix it up, store it in the fridge. So I've tried it and I really like it, but I'm biased. So if any of you want to try it and report back to me, I'd really appreciate knowing what you think of our sauce recipe. So to summarize our findings with the tahini, we found that increasing the water activity of the tahini paste can encourage salmonella replication, but that replication can be hindered or prevented by adding acid or cold temperature. It's important to remember, though, that the salmonella is not eliminated, it just doesn't replicate. So, in order to have beautiful salmonella-free tahini, we need to have cooperation from um, our international partners, both at the processing stage and the pre-harvest stage, to assure food safety. So if I did my job correctly, you now have a greater appreciation of the food safety risks associated with low moisture foods. The big takeaway is that there can be low levels of salmonella on low moisture foods. Despite these low levels, people are still getting sick. So what does that mean for dose response? That's the $100 million question that we're trying to work on. Also, these low levels have implications for detection in that we need to have larger sample sizes in order to increase the sensitivity of our method so we can detect these bacteria better. And the fact that so many outbreaks have been linked to salmonella and low moisture foods, it means that we really need to look at uh, the entire chain and see where we can add additional interventions. So that's all I have for you today. Thank you very much for your attention. And if there's time, I'd love to answer some questions.
Perfect. Any questions? Sorry, 1 second. Dr. Tamber, can you hear that if he's speaking, or should I pass on the mic?
I can't hear.
No, okay, I'll just pass on the mic to you.
Awesome. Yeah. Someone's super naive to salmonella and salmonella poison. What are the sources of salmonella poisoning? Like, where would it be introduced to these food products?
So salmonella loves to live everywhere. It's found in the intestinal tract of many animals, such as insects, birds, mammals. So in pre-harvest conditions, you can have droppings from animals. You can have salmonella in water sources. You can have salmonella in the soil. So it's pretty ubiquitous, I would say, in the environment.
Any other questions? Okay.
Hi, I was curious about the tween and the corn oil. How do you think that's interacting with, I think you said eugenol or whatever it is the antimicrobials are that are interfering? Because it's so interesting how you're working to improve the method. What do you think like the corn oil and the tween is doing? Is it just like surrounding that stuff or how is it? I don't know. How does that
Exactly.
work?
Exactly. You nailed it. So I think what's happening is the tween and the corn oil, they're forming like these micellar structures that are kind of sequestering the eugenol away from the bacteria. It's something that we really want to look at more to see if we can nail down that mechanism.
Any other questions? Actually, I have one. If you wouldn't mind talking a little bit more, like, have there been any like large scale transcriptomic studies or anything that have looked at the level of gene expression carrying between adapted strains?
So I'm going to paraphrase your question to be sure I heard it correctly.
Sure,
So
sorry.
you're so you're asking if there have been transcriptomic studies of salmonella in low moisture foods?
Yeah.
Okay. Yeah, there have actually. So those studies have largely used lab strains of salmonella, and so I don't really know how well they will generalize to these other serovars that we're seeing. But in general, the genes that are being identified are genes that you would expect to see in any type of stress adaptation activity, as well as transporters for um, various fatty acid types of molecules. So yeah,
Okay, interesting. Thank you.
you're welcome.
Anyone else? Are there any questions online? I haven't even looked.
I've got a couple of questions. Karen?
Take it away.
All right. Thanks. So it's Rhiannon Wallace here. I'm the Vice President of the BC Food Protection Association, and I also work not with Health Canada, but with Agriculture and Agri-Food Canada as a research scientist. So Sandy, I've got a couple of questions for you. When you were talking about your experiments with the seed powder and the biofilms, did you do any sequencing or knockout studies to see if there's any other factors, genomic features that are promoting those strains from surviving in the low water activity foods, other than just the ability to form biofilms?
So we haven't made any mutations. This work is still ongoing. We just finished doing some sequencing that we're in the middle of analyzing. Um, but I don't think we'll find anything because the genome of Salmonella tends to be quite stable. I think if you're going to see any differences, you'll need to look at that transcriptomic level of gene expression.
Right. Great. That makes sense. My other question was, so at the beginning of your talk, you talked about getting results initially taking 4 days, then you could get it down to a day. Do you ever see some of these culture-based methods being replaced with a metagenomics or quasi-metagenomics approach for pathogen detection?
I think these molecular methods, they're getting better. I think the big hurdle is the regulation. So right now in the regulations, you need to have a viable colony. Like the legal definition of a positive is to have a viable colony. So I don't think it's a matter of the science. I don't think it's the science that's the limiting factor. I think it's more the regulation and the policy side. They need to think about what are the implications of these molecular findings.
Great, thank you.
You're welcome. Thanks for your questions.
Any questions from the audience over here? No? Yeah, nothing online. Nothing online. So I think we'll leave it at that then. Thank you so much, Dr. Tamber, for taking the time. And I know it's late where you are, so extra thanks from us. Let's give one last round of applause for Dr. Tamber.
Well, thank you. Thank you again for the opportunity and enjoy the rest of your night.
Thank you so much.